El Recinto Universitario de Mayagüez presenta Foro Colegial TV, la revista informativa del acontecer del antes, ahora y siempre colegio, con un siglo de historia aportando a la educación del país.
Saludos y bienvenidos a Foro Colegial, la revista informativa del Recinto Universitario de Mayagüez. Soy Mariam Ludim Rosa, directora de la Oficina de Prensa del Colegio, y es un placer nuevamente compartir con los amigos y amigas que nos sintonizan en este espacio educativo del acontecer del antes, ahora y siempre colegio. Más de 100 años forjando profesionales de excelencia. La NOAA otorgó fondos al Recinto Universitario de Mayagüez para investigar las poblaciones del pez mero con una técnica de sonido. Sobre eso hablaremos hoy en Foro Colegial.
A continuación, la entrevista de hoy de Foro Colegial.
Saludamos nuevamente a los amigos y amigas en sintonía con Foro Colegial y para hablarnos sobre esta interesante investigación y otras que se llevan a cabo en el Instituto del Caribe de Arrecifes de Coral del Recinto Universitario de Mayagüez, se encuentra con nosotros su director, el doctor Appeldoorn, a quien le damos la bienvenida a Foro Colegial. How are you, professor, and welcome to Foro Colegial?
Thank you very much. I'm doing well. Thank you.
En primer lugar, él se va a estar dirigiendo a nosotros en inglés, se siente más cómodo en ese lenguaje. En primer lugar, quisiéramos saber un poquito más sobre el instituto en términos generales, ya que es la primera vez que el profesor nos visita. Professor, tell us a little bit more about the institute and its mission.
The institute was formed as a collaborative program between the University at Mayagüez and NOAA, and its purpose is to conduct and foment research that can be applied directly to management of coral reef and coral reef resources.
Mm-hmm. And who takes those research, those investigations?
The investigations are done in two ways. One is that we have received funds from NOAA to sponsor research programs, so we ask people to respond to our request for proposals. This is the work we would like to see done, and people who are qualified apply for those funds. The second way is that the members of the Institute itself can apply for other funds, not the funds we administer, but other funds to do research as well.
Who are the members of the Institute? Professors, students?
Membership in the Institute is kind of fluid because we have a staff, but beyond that, it is whoever we can attract who are the best people to do the research. So our family, if you will, of researchers can be people in the Department of Marine Sciences, other departments in Mayagüez, throughout Puerto Rico, or even outside Puerto Rico.
Mm-hmm. But mostly professors of the University of Mayagüez campus.
I'd say it's been about half and half.
Mm-hmm. So you have other professors from other universities that specialize in those subjects that you investigate?
Yes, we do. If we need a specialist that's dealing with aspects of genetics or soil erosion or some aspect that we don't have the expertise either within Mayagüez or Marine Sciences, we will go out and find that person.
So you were talking about that you have some funding from NOAA, and specifically these investigations related to the groupers, you have some funding from NOAA. What is about it?
The purpose of that investigation is to develop methodologies to monitor the spawning aggregations of groupers, to be able to use this information for management purposes. And the reason it's so important is that these fish come together in very dense aggregations at known locations, at known times, at least to the fishermen, and therefore most of the fishing for these species occurs at the time of aggregation. So if we can monitor that and monitor the population in a very simple way, we ought to be able to determine how our management measures are working or not working, as the case may be.
So the problem basically here is that those kinds of fish are overfished.
Well, in a historical sense, yes. If we go back 30, 40, even 50 years, the populations of these species was much, much higher. So there's been a lot of effort recently to bring fishing under control, and we think that those efforts are now having some success, but it's been a long time getting to that point, and we think that we have a simple methodology will start answering those questions about how well are the populations doing.
So how did you know that historic fact that they're decreasing the the populations of groupers. So many investigations before that?
Well, the Department of Natural Environmental Resources has been collecting fisheries statistics since the 1940s and 50s, and more routinely since the 1970s. So there's a long history of the catch records for these species, at least for groupers as a whole.
So now you're doing that with specific methods of the sound of the fish. Tell me a little bit more about that.
Well, the fish, actually just the males, make noise either to try and court females or to push away other males. So there's a constant noise that they make, a very specific sound, and each species has its own sound. As a consequence, when we listen, and they don't make the noise very loudly, so we have to listen very carefully with special instruments. If we listen, we can first of all detect where they occur by listening for the sounds, and secondly, by monitoring the sounds at those locations, we find that as the aggregation builds, first the males tend to come in first, and they'll start jostling with other males to kind of establish either territories or a dominance hierarchy perhaps within the group of males. And then as the females come in, they start courting the females. And as more females come in, they're increasing their vocalizations as they are either trying to attract females to themselves or push away males. So the bigger the aggregation, the more they're active they are, and the sound levels go up. So we have more or less a direct correlation between the sound levels, or the number of vocalizations that the males make that we record, and the total population density at that aggregation site.
So what kind of instruments you use to record those vocalizations? And you said it was difficult to know what kind of fish was doing that vocalization. How did you know?
Well, that's where the basic research comes in. We actually have to, first of all, find an aggregation, and then using cameras, either fixed or ones that divers use with hydrophones on them or underwater microphones. We actually have to capture on film the fish making the noise to know what that species' sound is. So we have done that for a number of different species, and as a consequence we can now monitor their behavior just by listening without having the cameras there or without having divers there.
So do you have this other instrument to record the noises, the vocalizations?
Well, we have two different versions of the hydrophones. One is one that we lower from the boat. It's a very simple mechanism. It's basically a microphone on a long cable, and the person on the top is listening with earphones, but also recording on a simple recorder what is going on. That's coupled with a GPS, so later on we can plot on a map the locations where we were hearing the sound. So that's one way to locate the aggregation. The more sophisticated instrument is basically the same microphone, but it's now in a housing maybe about this big. Most of that housing contains the batteries. The electronics are very small, but with the modern memory capacity, we can put that meter out before spawning starts, leave it there the entire time, and record throughout the spawning season, pick it up when we're done. And
how long? Months?
Months, yes. We would put out in November, and we pick up in late May or June, so we're actually scheduled to pick up
And there's no
meters.
harm that something happened with that instrument over there?
Yes, there is. Yes, there is. But we haven't had too many problems. We had one which we had put out just as a kind of a, we were searching for a new aggregation, we didn't know where it was, so we put one out. That was in the Mona Channel where the currents are very high. It was someplace where we didn't have any kind of soft kind of sediment to really hammer it into the ground, and the strong currents there actually knocked it over. And if we hadn't put a separate line on it as kind of an anchor line, we would have lost that into, I don't know, 100 meters worth of water.
Mm-hmm. Mostly the currents, there are on other curious creatures that go by the instrument?
Yes, there are curious creatures that go by it, but they don't seem to be bothered by it. In fact, some of the groupers, when the current's really strong, seem to actually hide behind it to try and get out of the current a little bit.
So mostly these investigations is being held in the Mona Channel, or there are other spots that you're listening to these noises?
All the locations that we have looked at so far have been in the Mona Channel, either at Mona itself or off the west coast.
So with what agency did you share your findings about the population of the grouper?
Anybody who will listen. So for agencies, we present that information through the Department of Natural Environmental Resources, either directly or through a special program called CMAP, which is a program designed to monitor fish populations. And so some of the hydrophones that we use actually belong to the CMAP program, and we are doing the monitoring for them in that program. We also obviously give our information to NOAA and the Caribbean Fisheries Management Council, which is based in San Juan.
So these microphones, these instruments allow you to have, let's say, like an inventory of the population?
That's where we're heading to. And
okay.
Since we have only been doing this for a couple of years, we're starting to learn the sources of variation in the data that we collect and the sound that we collect and the frequency at which the groupers come to their aggregation sites. That latter part is really variable from year to year. Some years they form a single large aggregation. Other years they'll form two smaller aggregations. Sometimes those are equal in size, sometimes they're not. So we're learning that kind of variation and how to anticipate that into our assessments.
In scientific terms, you share these findings on reviews, scientific reviews, scientific magazines?
Yes, we have already published a number of articles in scientific journals, both on the sounds themselves and the periodicity related to spawning, and most recently the one that was looking at the relationship of the sound levels to population levels or density of the fish at the sites. We also report them at scientific meetings through either regionally or internationally.
So far, what are the findings that you have in these 2 years of investigation?
Well, so far we have fairly strong correlations between the sound levels and the density of fish. I think that's probably the most important thing we found, because that really sets the basis for using this technology without having to have divers there, without having to have cameras there for long-term monitoring. We just put the hydrophone there, and it's there for several months. Every year we pull it out, and we can monitor the populations without having to deal with the problems of weather or shortage of personnel or even shortage of boat time and things like that, because we can put them all out at the same time, bring them all back at the same time. It's so much less expensive to do it this way. So if we can demonstrate that this is a procedure that works for all the species we're looking at, it would greatly enhance the information we have for management and reduce the costs at the same time.
Thank you very much for the information. Nos tenemos que ir a una pausa. Cuando regresemos vamos a seguir hablando con el doctor Appeldoorn sobre otras investigaciones que se llevan a cabo en el instituto. No se retiren, por favor. Saludamos nuevamente a los amigos y amigas en sintonía con Foro Colegial. Estamos conversando con el doctor Richard Appeldoorn. Él es director del Instituto del Caribe de Arrecifes de Coral del Recinto Universitario de Mayagüez. Durante esta primera parte hemos estado hablando sobre una investigación. La mayor parte de las investigaciones de ellos están subvencionadas por NOAA. También otras agencias han subvencionado sus investigaciones. Esta investigación en particular, de la que estuvimos hablando en el primer segmento, está auspiciada por NOAA. Específicamente se están buscando las poblaciones en desove del mero, pero es el Red Hind, ¿verdad? Grouper, que es el mero cabrilla. Específicamente ellos están investigando sobre esas poblaciones porque a través de las décadas esas poblaciones han disminuido por la sobrepesca. La técnica que están utilizando para saber qué cantidad aproximada hay de estas poblaciones es a través de la grabación y el sonido que hacen estas especies, específicamente el macho cuando están en el periodo de apareamiento. Y es bien interesante, ya el Recinto Universitario de Mayagüez lleva unos 2 años con esta investigación, la que ya se ha publicado en revistas científicas, y ellos pues quieren continuar con esta investigación para también darle esta información información a otras agencias que están relacionadas con el control de la pesca. ¿Resumí bien lo que nos dijo, profesor? Did I summarize well? Did I pass the class?
Yes, you did.
Yes, I did. Well, we keep on talking. So, professor, in these investigations, what's the role of the students? You have many graduate students that work in these investigations.
Yes, we do. And the work that we do is fairly labor intensive because we're trying to connect the vocalization data with what the behavior of the fish is and the abundance of the fish. So there's a lot of diving involved at the aggregation sites. This requires usually a team of about 4 divers when we go out, so we can do replicate dives during the day. And we also need somebody to actually listen to all the recordings so that we can classify which species are making noise and how much sounds they are producing.
I was wondering if they dream about those sounds after a whole day listening to them.
I believe they do, yes.
Yes, yes. Professor shared with us, compartió con nosotros algunos de esos sonidos y son bien interesantes. Ellos varían con relación a la intensidad y con relación al pitch del sonido, pero pues tiene que ser un especialista el que los escuche para conocer la diferencia. You have any other specialists that have helped you with this investigations outside the university?
Yes, actually the work started with a grant from the Institute to a researcher in the University of South Florida who was a specialist in fish sounds. And it was his idea that perhaps we could monitor the aggregations by listening to the fish. At that point, we didn't know what they looked like. He was awarded a grant from the Institute to do that. I was lucky enough to have some people, graduate students at the time, in my lab who could work with them because the work was being done at Mona and we could logistically facilitate that. And through that connection, we kind of developed the expertise. So when they were able to actually record the sounds of the fish and have some kind of baseline about where they are and what time of year that they're aggregating, we were able to then base our subsequent investigations focused on assessment on his initial work. And so, he's been involved throughout the project.
So the importance to have a good population of this kind of grouper is because of the reef, the coral reef population also? They are related?
Well, there are 2 reasons. Obviously, one is food supply. The mero, cabrilla, or red hind, is one of the most popular fish and most abundant of the commercial fish species that are being fished, so we want to maintain that in as healthy population as we can. But there's a secondary role for this species and even the larger groupers that are even more rare at the moment, is that they control the populations of other fishes, which then control the populations of invertebrates and other creatures on the reef. So there's what we call a top-down control of the population levels, including eventually herbivores, which have important roles in controlling the amount of algae that are growing on reefs, and they are competing with the corals. So if we want to have healthy coral reefs, we want to have lots of herbivores. So we want to, if there's a kind of a chain reaction here, you want lots of herbivores, then you want few predators on the herbivores, which means you want lots of predators on those predators.
So what are your plans on the short term related to this investigation?
Right now we're working with another species, which is the Nassau Grouper, or mero, and that's a species that most likely will be listed very shortly in the Endangered Species Act. So anything we can learn about that species and put into place management measures that will allow it to come back will be really important. And this is a species that was probably the most important, certainly the most important grouper species and probably the most important fishery species in Puerto Rico going back to the 1900s.
So now that we spoke about the coral reef, let's turn out to other subject. You do many investigations. We will need some programs to talk about them, but I would like to talk now briefly about the cruises that you have investigating the coral reef. Can you tell me a little bit more about that?
Yes, this is also work that was funded by NOAA, and it was really at their initiation that we started investigating coral reefs at depths between 50 and 100 meters, so it's about 150 feet to 300 feet. To do this required developing a whole new methodology, and so we developed the capacity to put divers in at those depths using rebreathers and mixed gases, so they're using oxygen, air, and helium, so it's called trimix. So it's a very special technology that allows them to go that deep, and we've been working that now since about 2007. First in the area off La Parguera, we had a long study there, and then based on that we were able to put forth a series of questions on what might be happening around at least the northern Caribbean, and we were able to get funding from NOAA for 3 cruises to go look at these special what's called mesophotic coral ecosystems. Mesophotic meaning half light because they are very deep.
So what's happening with the coral reef?
Well, at those depths, things are looking actually quite good. We've learned a lot about where we might find really nicely developed reefs because they are patchily distributed, just as our shallow reefs are sometimes here, sometimes there, but not everywhere. We've learned a lot about the species compositions, and what we didn't know was what kind of variations we were having in their distribution depending on the geomorphology. You know, what is it like when you get outside of La Parguera, for example? Differences in conditions in terms of, as we were mentioning, fish populations. So there are certain places where fishing is restricted, and do we see more fish deep in those areas or not? We are looking to see if there's variations in the factors that control the distribution, both on a large scale and a small scale.
So are you willing, or are you looking forward to have more cruises in the future? I know that the agencies all have budget problems, but you know, you make some proposals.
We're not immune from the budget problems. Mm-hmm. Yes, we're continuing to write proposals. We do not have a cruise scheduled at the moment. What we are focusing on is working up the analyses of the data that we've already collected. What I didn't mention, one of the major things I didn't mention was the use of genetic techniques to try and understand the dynamics of these populations, particularly how they are connected with either shallow reefs or other reefs at distance, say in the Virgin Islands or eastern Puerto Rico, western Puerto Rico, and Mona. So using genetic techniques, we can look and see how the genes are differing, which may reflect whether there is connectivity through presumably larval dispersal between these areas or not.
Can you tell us a little bit more, what do you referred when you said genetic techniques, so we can understand?
What we actually do is try to make a map of the DNA, not for the entire set of DNA, but for certain parts that we can get that are specifically selected because they vary at a rate, say spatially, that will give us information about whether these two populations are sharing the same pool of adults and juveniles, or whether one group is separated from another group, maybe by currents or distance. So their DNA tends to be different.
So these findings you also have published on scientific journals?
These findings are still being worked up. A couple of them are just about ready to be sent to the journals. So, yeah,
and Professor,
it's a very exacting
in general terms,
procedure.
you told me that in these cruises, because of the depth, you found that the coral reef didn't have those problems that you have told us before, like the bleaching or some problems like that. What are the main problems with the coral reef?
Well, in shallow coral reefs, we have basically two problems. We have inputs from the land or overfishing, activities that we are doing locally to upset the balance of the coral reefs. We're having too many nutrients come out, either because of sewage or fertilizers or things like that. Too much sediment because we're not really controlling the amount of erosion when we do development or farming or those kinds of activities. The other class are really global. So we have temperature increases in global warming, and we also have acidification. As CO2 is put into the atmosphere, that is absorbed by the ocean. That keeps the CO2 in the atmosphere lower, but it raises the acidity of the oceans, and they will eventually make it more difficult. They already are making it more difficult for organisms that use calcium carbonate, such as corals, mollusks, and lots of other organisms, to make their shells.
So there's a lot to investigate.
There's a lot to investigate, and those deeper corals are going to be some of the ones that are going to be impacted first, because there's a temperature effect, and the cooler the temperature, the effect will be felt earlier.
Thank you very much. We run out of time. Thank you very much for being with us and sharing all these magnificent investigations that you do in the Institute.
My pleasure.
Gracias, amigos, por una vez más estar en sintonía con Foro Colegial. Recuerde que historias como estas las puede acceder en nuestro portal www.uprm.edu. Será hasta la próxima.
Hasta aquí, Foro Colegial TV. El espacio del acontecer del Recinto Universitario de Mayagüez, producido por su oficina de prensa. Para comentarios o información adicional, puede escribirnos a prensa@uprm.edu. Gracias por su sintonía.